So, yesterday we talked about free energy and how free energy affects our reaction, as well as how to calculate free energy. And at the end of the day today, and the beginning of the day on Monday, we are going to go, we're going to have some practice with those actual calculations. We got a little practice sheet for us. But today, we are going to expand our metabolism vocabulary with endergonic and exergonic reactions, specifically involving ATP. So, my goal for you is to be able to calculate Gibbs free energy, as well as use that calculation to determine whether or not that reaction is endergonic or exergonic. Whether it will occur spontaneously or non-spontaneously. So, starting us off with why we need these words, we can use those free energy equations. If you remember, free energy is, if I can get my little pen started. Yes. We have a symbol. Nope. Free energy. We use a capital G to represent that. And when we want the change in free energy, we use the Greek letter delta and that G. So you should have the G and delta G stuff in your notes already. I just kind of wanted to remind a little bit. And we can use that calculation to determine whether or not something is endergonic or exergonic. I'm going to talk about why or what the difference is between those two. So, does anybody have any specific questions about yesterday before we move on? Yes. It is not. I can pull the equation sheet up. Go on an adventure together. Aha, this is the equation sheet. We've got chi-squared, standard deviation, a bunch of prefixes, mean, median, mode, Hardy-Weinberg, which we'll talk about next semester, volume, water potential, and growth rates. So the Gibbs free energy equation is not on the AP or on the equation sheet. So we might have to commit that one to memory. Luckily, it's not the most intensive equation that we have to deal with, and I'll bring that back to you. A lot of the times, though, if you'll use it, they will give it to you in the question, but because it's not on the equation sheet, it's not guaranteed. So we will definitely have a lot of practice, and we'll review that right before the AP exam as well. So a little free energy review. You don't have to write this down because this is what you had in your notes yesterday. If you want to write it down, that's great. Remember, free energy is the portion of a system's energy that is available to do work, and we represent that change with that delta G symbol. So, the energy available to perform work. So that's our little review from yesterday. Now, I'm going to get into the new stuff. So, an exergonic reactions will proceed with a release of free energy. Think exergonic exhale, release of energy. Exergonic. And those have a negative delta G. So if you have an equation and it has a negative delta G, you know that equation or that reaction is an exergonic reaction. Exergonic reactions, or reactions with a negative delta G, will proceed spontaneously. But I've got a little caveat about spontaneity. When we say in chemistry and physics that a reaction occurs spontaneously, we mean that it is energetically favorable, meaning it like the reaction prefers, or the reaction naturally is pushed towards that release. This does not mean that it will occur rapidly. So just because a reaction is reaction is exergonic, just because it happens spontaneously, does not mean that it will happen really fast and all at once. You remember the glow stick from yesterday, right? That reaction takes place over the entire course of that glow stick being lit up, right? Because it is the reaction that is releasing the light. So that is a very long form release of energy, because that energy is released over the course of however many hours that glow stick is useful for. If you've ever had one of those like hot hands hand warmers, right? When you start mixing those two things together, it releases heat. I'm a naturally cold person, so I usually have a bunch of those on hand around the winter time. Those things are an exergonic reaction. Once you mix those reactants together, they release energy in the form of heat. That heat doesn't all happen all at once. It's not like you mix it up and then all of a sudden it releases all of its heat all at once. You burn yourself and it's done, right? It's a slow release over time. So even if the reaction is slow, it can still be spontaneous. It doesn't mean that it's all happening all at once, and it doesn't mean that it's happening randomly. So if those two reactants are mixed together, it's not like they wait, and then just go. So spontaneous means energetically favorable. It will proceed toward that release of energy at a specific pace, and almost instantly as they start moving on. That's what spontaneous means in the world of biochemistry physics. Questions here? Okay. I have a little chemistry biology caveat for you. So, when we say that we break bonds to release energy, right? Usually we say that it takes energy to make bonds and you break bonds and get that energy. And we'll even talk about how when we use ATP, we release energy from that breaking of that bond. But here's the thing. The breaking of bonds does not release energy. Like full stop. Does not release energy. When we talk about the energy stored in chemical bonds, we are referring to the potential energy that will be released when new bonds are formed after the original ones are broken. It actually takes energy to break those bonds. So, when we talk about releasing energy, when we break a bond, we are not talking about that bond having energy in it. We are talking about the new potential energy that exists as those bonds are formed after the original ones are broken. This is the case as long as the products are of lower free energy than the reactants. Because if the products, the end goal, has a lower amount of free energy than the products, we have that extra, that released energy. So this is kind of a little caveat, because we will say that when bonds are broken, energy is released. But that's just because that's a shortened version of this like skipped step. We're kind of summing it all up in that bonds broken, energy released. But in actuality, it's not that those bonds are holding energy in and of themselves. It is that when we break those bonds, they will have more energy in them, or not in them, but the potential energy of the new bonds formed is more than the energy in those products. I think that's the sentence I wanted to say. It's kind of rambly towards the end. little physics caveat. That was exergonic reactions. The opposite of an exergonic reaction is an endergonic reaction. N in. So exergonic, endergonic. Endergonic reactions absorb energy. They have a positive delta G. They have a positive change in free energy as a reaction. These are non-spontaneous. You need to put energy into them to get them started. The magnitude of delta G, whether that delta G is 1 or 101, it will still need an energy input. But that delta G of 1 will need less of an energy input than the delta G of 101. The magnitude of that delta G tells us how much energy we need to get the reaction started. So, a low positive number takes a lot less energy to get going than a high positive number. Yeah. So when a delta G is a low positive number, it just means there's less energy required to get that reaction started than when delta G is a high number. For example, a delta G of 1 takes less energy to put into that reaction to get it started than a delta G of 50. So that is an endergonic reaction. How big that delta G is, that positive delta G is, tells us how much energy we need to kickstart whatever reaction we're looking at. Doing okay so far? A lot of chem, a lot of physics starting us off. All right. So we can represent these graphically. So these graphs, we've got an exergonic on one side and an endergonic on the other. Essentially, and we've got free energy down this axis and time down this axis. Of course, we're not going to be able to write on this right now. Why would we be able to? There we go. So, in an exergonic reaction, if we are going to draw this out, we have our little axes, time, energy. Our reactants start higher, we get a little bump, and we have our products lower. This difference is our delta G. That is an exergonic reaction because we are releasing energy. There is a big energy difference here, a negative energy difference with the exergonic reactions. With the endergonic reactions, our reactants, the things going into our reaction, are lower than our products. So our delta G, our change, our difference between the two, is going to be positive. Because we have to put in all of this energy in order to get to where we need to go. Whereas this one over here, we can lose energy and get it where it needs to go. And that energy has to go somewhere, so it's released. And these little things down here, it's stuff you already have, right? Energy is released in an exergonic reaction. It's spontaneous. Our delta G is negative. An endergonic reaction, energy is absorbed. It's non-spontaneous, and our delta G is positive. I don't know why it helps. I don't know why I started doing it. But whenever I think exergonic and endergonic, I usually think of some sort of physical movement. So exergonic is out, endergonic is in. It helps me remember which ones release energy and which ones take in energy. So exergonic releases energy, endergonic takes energy in. And a lot of the times we can represent this graphically. Next week, we will talk about activation energy, the energy needed to get a reaction started, specifically when we talk about enzymes. And we will probably revisit these graphs again talk about what activation energy is. So, exergonic, endergonic reactions, we can see physically where our energy differences are. Questions so far? All right. Equilibrium. So, our universe kind of pushes all reactions toward equilibrium. That's the goal. Reactions in an isolated system, remember, isolated systems are not going to exchange anything with the environment. In an isolated system, eventually they will reach equilibrium. Equilibrium is when everything is all evenly distributed. There's no major reactions going on that are creating like a difference between parts of this reaction. Now, here's the thing. Metabolism is never at equilibrium. Metabolism is never at equilibrium. And this is that defining feature of life. And I will put in my notes to find that little podcast clip so we can start out nice and easy on Monday. So, we, in our bodies chemically, are never going to be at equilibrium. There's always going to be an imbalance of where those reactions get going. But in an isolated system, if we are running models, that is what everything is driving toward. Now, all of this gets us to how a cell uses energy, right? We got all this foundation, and now we start getting into the bio in this biochemistry. So, there are 3 big types of work that cells have to do. 3 big things that cells need energy for. The first is chemical work. There are a lot of endergonic reactions, a lot of anabolic reactions where we are building things up. Those chemical reactions take energy to get started. Those endergonic, those anabolic reactions need energy to get started. So we call that chemical work because we need energy to do that work. The second is transport. We talked about the electrochemical gradient, right? That electrochemical gradient takes work to maintain because we are actively moving things against their concentration gradients. As we move things against their concentration gradients, it takes energy. That is active transport. That active transport is one of the things that cells need energy to do. And finally, there's actual mechanical kinetic work, right? Cilia gotta move, flagella gotta move, our muscle contractions, that takes energy. Same with the movement of chromosomes. When meiosis happens, we are physically separating chromosomes to either parts of the cell. That physical separation takes energy. That movement takes energy. They are not naturally pulled to either side. That cell has to work toward doing that. So there are 3 big types of things that cells need to do, that they need energy for. Now, if we need energy for this, where does that energy come from? What's our energy source in cells? That is ATP, or adenosine triphosphate. Adenosine triphosphate is what powers all cellular work, nearly all cellular work. And it does so by coupling endergonic and exergonic reactions. If we get two reactions where one releases energy and one takes in energy, and we put them together, the goal is to use the energy released by the exergonic reactions to fuel the endergonic reaction, instead of that just being released into space. Right? We're going to harness that power. And when we talk about cellular respiration in a little bit, I mean like a week or two, we will talk about how ATP is formed. So, this is ATP. ATP is made up of a ribose sugar, an adenine group, and a chain of 3 phosphate groups. And we are going to draw this. We are going to have this in our notes. So, we have, if I can get down here, an adenine, which is, I believe, 2 benzene rings. Oh, there's nitrogens in there. They're not benzene. Oops. Okay. This is our adenine. If you want to throw the little elements in there, you can. You definitely do not have to. This is the same adenine that is in our DNA. Those are the A's in the ATGCs. Same thing. We're just using it for something else. Adenine group. We then have a ribose sugar. So, coming off of this, we've got a little pentagon. This is our ribose sugar. That's looking a heck of a lot like a nucleotide. When we talk about nucleotides in DNA, we'll talk about how we form these connections, why this is so stable. Okay, we'll get into it. But instead of instead of 1 phosphate group, we have 3 phosphate groups. So, coming off of this ribose sugar, we have phosphate group number 1, phosphate group number 2, that one got a little small over here, phosphate group number 3. From the very, 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 very beginning of the year, we talked about all of these special groups that are added on to different organic molecules. This was one of them. We've got 1, 2, 3 phosphate groups. This is ATP. It is this molecule that powers every single big energy requirement in our bodies, in life, in living things, not just humans. This molecule right here powers everything from the plants outside to the earthworms squiggling around to you and me. Very important molecule. A lot of the times you will just see the shapes written out. Sometimes you'll see these written out. I had you do the actual phosphate group bits just because it is these bonds that get broken that release energy. And being able to see those is important. Just like whenever we talked about, what did we talk about? Oh, amino acids. When we talk about in protein synthesis, where amino acids are joined together, I think we had a Fourier response AP exam question last year where they were like given the chemical structure of an amino acid and they had to figure out where one amino acid stopped and one amino acid started. So some of these chemical groups are important to recognize, and ATP is one of them. So when we break one of the bonds, my circles didn't stay where they needed to be. When we break one of these bonds, we do so by hydrolysis. Does anybody remember what hydrolysis was? Where we used hydrolysis in the past? Come on. There you go. Circles are a little small, but it's fine. There we go. So, when we break the bonds in ATP, we do so through hydrolysis. Remember, hydrolysis is hydrolysis. It is using water to break things apart. It was one of the things that we used to break apart our monomers when we talked about our macromolecules. So, there we go. When we do this, we use ATP and water. We break that apart. We get ADP and this little phosphate. It's a P with a circle and a subscript I. We call this an inorganic phosphate. One of the few inorganic molecules that's super duper important. Not few, but like one of the big inorganic molecules we hit up a lot. More chemically, it is HOPO3 2-. But usually we just call it an inorganic phosphate. This reaction, adding water to an ATP molecule and breaking it up into ADP, or adenosine diphosphate, because we've lost the phosphate group, has a delta G, I think that should be negative, of 7.3 kilocalories per mole. I believe that should be negative. Let me double check. It should be, like, logically. Yes, negative. Do it here so it's there for good. Negative. Because it is an exergonic reaction, it's going to release energy. It's going to have a negative delta G. And that negative delta G is negative 7.3 kilocalories per mole. So for every mole of ATP that goes through this reaction, we are going to release 7.3 kilocalories of energy. Now, when we talk about this bond, these phosphate group bonds, we call them high energy bonds. But that's not because the energy in the bonds is super high. It is because the ATP and water have higher energy compared to the ADP and the inorganic phosphate. It's that little caveat we talked about earlier. So this hydrolysis, this using water to break up this molecule, is like the reaction that gets stuff started. So, how does hydrolysis work? We do this in a lot of different ways. For example, enzymes. Enzymes are super important. they will use the energy, that negative 7.3 kilocalories per mole, from hydrolysis to directly drive endergonic reactions. If they do this, our delta G of the endergonic reaction has to be less than the delta G by the exergonic reaction, right? Because we have to be able to have enough energy released in order to get the other energy, or the energy needed to get the other one started. Monday and Tuesday are going to be all about enzymes. So, this is how the hydrolysis works. And I've got a specific reaction we're going to draw out just so we have like a baseline example of how this coupling works. So, we're going, R. So, if we have a reaction that has a positive delta G of 4, right? If we need 4 kilocalories of energy to get that reaction started, and we couple it with this hydrolysis of ATP that releases 7.3 kilocalories, we can do that because that 7.3 is enough to cover that 4. But if we had an endergonic reaction that needed 13 kilocalories of energy, we don't have enough energy released by that hydrolysis of ATP to cover the 13 needed to get that endergonic reaction started. This is how we can kind of use those delta Gs to see if we can pair reactions together. So, when we use ATP hydrolysis, a lot of the time, we are going to directly transfer that phosphate group to another molecule. So instead of getting that like loose inorganic phosphate, we're just going to chuck it on something else in a process called phosphorylation. When we add a phosphate group to something, we call that phosphorylation. What this will do is create a phosphorylated intermediate. That phosphorylated intermediate is going to be the key to getting some of these reactions started. I've got an example. So, instead of just using the energy from the release of that ATP and that organic phosphate, just parsing that energy in another way, a lot of the times, in order to kind of make sure that energy stays where it needs to go, we will transfer that phosphate group to another molecule directly and use that phosphorylated intermediate to keep the reaction going. That's why it is called an intermediate. It is not a product, it is a step in our equation. All right, so I've got an example reaction I want us to write out. We are going to take glutamic acid with ammonia and make glutamine. So we're going to write this out together. We got a little triangle. This is glutamic acid. And we are going to add ammonia. And we are going to get glutamine. This is just an amino acid. These are glutamic acid and glutamine are amino acids. You need to make amino acids sometimes. So this is that process. The delta G of this reaction is plus 3.4 kilocalories per mole. The delta G of this reaction is plus 3.4. Is this reaction endergonic or exergonic or endergonic? Endergonic, right? We need energy to get this started. It needs to take in energy. So what we're going to do is we are going to create or couple it with ATP. So here's what's going to happen. We're going to still start with our glutamic acid. But instead of going straight into the ammonia, we are going to add ATP. This will give us that phosphorylated intermediate. We're going to take one of those phosphates off of ATP and we are going to add it to the glutamine. So now we've got this glutamic acid with that phosphate group on it. This is going to be our phosphorylated intermediate. We also get out of this ADP. ADP because we lost the phosphate. So instead of triphosphate, it's diphosphate now. One of our phosphates is over here. So ATP, what it did was it went from to, hold on. It gave one of these phosphates here, and now we are left with two of them. We got our next step in the reaction. This is where our ammonia comes into play. That ammonia is now going to be added to that phosphorylated intermediate. So we've got our phosphorylated intermediate, that NH4, that ammonia is going to be on there. So we get our glutamine back. And we have that other phosphate, that inorganic phosphate. Our reaction over here has ADP twice. They just carried it over. So the ADP is still just chilling there. So our delta G for this first reaction was negative 3.4. So our delta G for the glutamine was positive 3.4. And we know that the delta G of ATP, of that ATP phosphorylation, is negative 7.3. So when we do both of these reactions together, our total delta G, or our net delta G, is just going to be those added together, is negative 3.9. Is negative 3.9 exergonic or endergonic? Exergonic. So when we were just looking at the synthesis of glutamine, this one was endergonic. This one was not going to take place on its own. We needed to figure out a way to get that reaction started. And we did that by coupling it with that hydrolysis of ATP. Kind of looks like the scribblings of a madman. Do we get where these are coming from? Does anybody have any questions about this process? All right. So we can also, my picture cut off my title slide. We have to regenerate ATP, right? ATP isn't a one and done kind of thing. We can build it back up. It just takes energy. That plus 7.3. We got to get it back in there. It's like a battery. It's like a battery that we can charge and recharge. It's like a battery we can charge and recharge. So, we're going to start out with ATP. Our phosphate group, yada, yada, yada. We've got our adenine, ribose, sugar, and our 3 phosphate groups. So you want to put little P's in here. That's great. This is ATP. This is where our energy lives. This is where our energy is. We can go through the hydrolysis of ATP by adding water to break it up. This is going to release energy. And we are going to get ADP, what's a D? ADP, and that inorganic phosphate. This is our dead battery. It has the potential, not potential energy, but it has the, it is possible to make it become energy again. But we need to do something to it. We need to phosphorylate it or dehydrate it. So we do that. When we do that, we release water. This is going to require energy. And we will get right back up to ATP. This release of energy, 7.3 kilocalories per mole. Your unit. This energy requirement, 7.3 kilocal per mole. This reaction here has a negative delta G. This reaction here has a positive delta G. Yes. It's a lot of different small names. We can call it phosphorylation because we are adding a phosphate group. We can call it a dehydration reaction because we're losing water. Usually, we can just say the regeneration of ATP. And that process is endergonic. And the process of releasing that energy is exergonic. Questions here. All right. So, we only have about 5 minutes left. I have a Gibbs free energy sheet linked on Canvas that I was going to pass out because I expected us to have like 10 to 15 minutes left, but writing always takes longer than I expected to. So we are just going to do that on Monday. Don't worry about it. The due date on Canvas is until Monday anyway. It'll be kind of our ease into the week. Does anyone have any questions about what we talked about today? All right.